In this presentation, we will talk about adaptations to life in water. There are 5 general topics here. First, the properties of water. Second, the adaptations of organisms to the density and viscosity of water. Third, osmoregulation in aquatic environments. Fourth, gas exchange in aquatic environments. And fifth, heat balance and adaptations for different temperature fluctuations. So water has a lot of unusual properties in the physical world, and its uniqueness is one reason why living things probably will always evolve in association with water. First off, water is thermally stable. It has a high heat capacity. It stores much more heat than air or soil. And as a result, it can modify environments, terrestrial environments that are close to water, by buffering that land from extreme temperature swings. Because if it gets hot, the water absorbs quite a lot of that heat without heating up that much itself. And if it gets cold on land, the water will give up a fair amount of its heat without cooling itself. Water reaches its maximum density at 4 degrees centigrade, which means that ice floats, and that is very unusual in the physical world. The density of water, though, means that living organisms often have to have specific adaptations to live in a water column and to find the area within the water column that is optimal for their activities. Third, water is viscous. It resists movement because of the cohesion of water molecules in a body of water. They form hydrogen bonds and stick together, and so it's harder to move through water than through air. Another feature of water is that ions easily dissolve in water, and they are readily available to organisms in ways that are not true of terrestrial habitats. However, the flip side is that gases tend to be in lower concentrations in water than in air, and that can limit organism activities. Finally, pH can greatly affect the function of organisms in aquatic environments. Organisms function best at neutral pH and are progressively impaired as pH gets low, that is acid conditions, or high, that is alkaline conditions. What can we say about major problems in adapting to life in water? Well, density and viscosity impose similar challenges and require somewhat similar solutions, because organisms in water may need adaptations to move effectively and to sink or float to the appropriate level. Osmoregulation is important because solutes in water may be more or less concentrated than in tissues, and the tissues of living organisms are mostly water. So just by osmosis, water may move in and out of organisms inappropriately, or solutes may move in and out of organisms in ways that impair their function. Third, gas exchange. It is important for organisms to be able to extract gases from water, and they need special adaptations to do this given the low concentration of gases in water. And then finally, the thermal environment of water is quite stable. Now that produces consistent selective conditions, and it's quite easy for organisms to be modified by natural selection to have a sort of general rule of thumb adaptation that is suitable for a particular thermal environment in water, because water is so stable. So let's take these one at a time. First, let's look at adaptations to the density of water. Density makes it difficult for organisms to move, and because organisms have a density of their own, they will tend to find a level within a water column that matches their density. However, that may not be where they really want to be. They may want to forage at a level in the water column that's quite different from their own density. So organisms need to be able to modify their density in order to move up and down from less dense upper layers of water to denser lower layers of water. So in this little picture over here, we have oil droplets within an alga that allow that alga to rise in the water column because, of course, oil is less dense than water. The alga can sink simply by metabolizing some of these oil droplets and converting them into other forms of matter, in which case it will tend to sink as it gets denser. Fish can't do that sort of thing, but they have swim bladders, and they fill them with gases that they extract from the surrounding water, and they can modify their own apparent density in a water column by inflating the swim bladder to rise, or removing gases from the swim bladder in order to increase their density and sink. So, water is also viscous, and that makes it difficult to move through water. Now, there are two different challenges. One is for large organisms that may want to move, and large organisms generally require streamlining for efficient movement. So fish have a particular shape that reduces the drag on their bodies if they need to move in the water. On the other hand, tiny organisms may want to stay in water, and they may maximize the drag in order to stay in one place, as this little larva has done here, by having all these projections off of its legs that those projections adhere to water molecules and tend to keep it in one place. And that prevents these little guys from sinking to the bottom of the ocean. Okay, second thing is salt balance. This is an incredibly important issue in aquatic organisms. And there's going to be some vocabulary we have to deal with here. So solutes are substances that are dissolved in water, and that affects the osmotic potential of water. Solutes that are in high concentrations in water tend to attract water from areas of low concentration in order to balance the overall concentration. So you get movement of water molecules from areas of low solute concentration to areas of high solute concentration. Similarly, solutes can themselves move from areas of high concentration to low concentration through diffusion. Now, organisms are bounded by semipermeable membranes, and the movement of molecules through semipermeable membranes is osmosis. Semipermeable membranes are very important in reducing the unwanted loss of solutes from organisms, but that imposes its own challenges, because if you prevent solutes from moving across a membrane and then you do want them to get from one side to the other, you may have to invest energy to actively transport them from one side to the other. Now, the gradient between solute concentration on one side of a semipermeable membrane and on the other determines the strength with which water is pulled from the low concentration side to the high concentration side. And that is called osmotic potential. So here's some vocabulary we really need to deal with here. So osmotic gradients are characterized by comparing the two sides of a semipermeable membrane. In an isoosmotic situation, solute concentrations are more or less equal on both sides of a semipermeable membrane. If they are unequal, then one side of the membrane is hyperosmotic. That's the side of the semipermeable membrane having the higher solute concentration. Water moves toward the hyperosmotic side through the membrane. Solutes, if they are allowed to, will move away from the hyperosmotic side through that membrane. On the other hand, the opposite side of the membrane in this situation is called hypoosmotic. And that's the side that has the lower solute concentration. Water moves away from hypoosmotic areas to hyperosmotic areas. Solutes move from hyperosmotic areas to hypoosmotic areas. Now, it's very important to realize that these terms describe comparisons. They do not describe conditions of some organism. An organism is never hyperosmotic. It is always hyperosmotic in comparison to the surrounding water, or it is always isoosmotic in comparison. A fish can have a particular solute concentration and be hyperosmotic to fresh water and hypoosmotic to salt water. So never, never, never use these terms to describe the characteristics of an organism. Always use them to compare organisms to the surrounding water. Now, osmoregulation has opposite challenges in freshwater and saltwater. So let's examine what might be the challenge in freshwater first. So here's a freshwater fish, and here's the relationship of water in the fish and water outside the fish. Water has fewer solutes if it is fresh than the organisms that live in it. So that means that the organism, the fish, is hyperosmotic compared to the surrounding hypoosmotic water. Water will tend to move into the fish to equalize the solute concentration and dilute the blood and cellular fluids of the fish, making it very inefficient and even possibly killing it. So freshwater fish have to have adaptations to remove excess water. Furthermore, they can't simply seal themselves off from the water because they need to get gases from the water for respiration. So there's always a semi-permeable membrane exposed. Sometimes it's in the gut, but in addition, there is always water gain from the gills, which don't have a choice. They have to be exposed to the water. So water moves into the blood of a fish through the gills constantly, and constantly has to be removed. The fish do this by producing urine, and they produce a very dilute urine in order to get rid of the water and preserve as many of their solutes as possible. The situation with solutes is exactly opposite. There are more solutes in the fish than in the surrounding water, so solutes want to leave the fish and move into the surrounding water to equalize the concentration gradient. That means that fish have to invest a lot of energy to actively transport solutes out of their urine back into the fish to conserve them. For saltwater fish, it's exactly the opposite problem. Seawater is hypertonic compared to fish, and the fish are hypotonic compared to seawater. So the salt concentration of the oceans means that there's an osmotic gradient continually pulling water out of the fish and dehydrating it to the point of death if they don't take action. So how do fish prevent this from happening? How do they stop themselves from losing their water? Well, they're going to have to lose some water through the gills. So they've got to drink water to replace the water they lose through their gills, and then they have to invest energy in producing concentrated urine that is actively transporting the solutes they take in into the urine before it is expelled from the body and retaining as much of the water as possible. So both freshwater and saltwater fish use the same system for osmoregulation, but they do it in exactly opposite ways. Freshwater fish try to conserve solutes and get rid of excess water. Saltwater fish try to conserve water and get rid of excess solutes. So in terms of the solutes, the water has more solutes than the fish. The salts want to move into the fish and impair its functioning, and so you have to actively transport a lot of solutes, especially sodium chloride, into the urine and make it a very, very concentrated urine to get rid of that excess salt. So this is not the only way in which organisms can do this. Most organisms that live in water, most complex organisms like fish, do exactly what I described. However, there are some alternate pathways. So over here you can see what a mammal does. A mammal consumes protein. It makes ammonia from that protein, and it excretes the ammonia directly without converting it to uric acid or urea, which are less harmful byproducts. On the other hand, sharks solve the problem of osmoregulation by converting ammonia in their food to urea, and they retain the urea in their bloodstream. Well, that's an extra solute, and it raises the osmotic potential of the blood so that it is isoosmotic to seawater. There's a problem, though. Urea is toxic. So in order to protect themselves from this toxic urea, they also synthesize a molecule called triethylene oxide, and that shelters their tissues from the harmful effects of urea. So there's more than one way to tolerate these osmotic problems. Sharks exemplify one way, and bony fish exemplify a very different way. Now, osmoregulation in plants is equally difficult in seawater. If you grow in a salty environment, then you are constantly facing the same problem. The salty water is going to be hyperosmotic compared to your tissues. Salt is going to want to move into your tissues, impairing your function, and it's going to try to leach water out, dehydrating your tissues. So, plants like mangroves that grow in saline areas don't necessarily try to avoid absorbing the salt. They do try to maintain high concentrations of organic solutes in their roots, like phosphate, nitrate, to raise the osmotic potential of their roots to lower the amount of salt that is able to diffuse in. They can't prevent salt from diffusing in altogether, so they excrete salt on the surface of their leaves through special glands to maintain a workable osmo-regulated system in their tissues. Let's move on to gas exchange here. Okay, gas exchange for photosynthesis is really limiting for aquatic organisms. Plants need CO2 for photosynthesis, and CO2 is not present in seawater or in any water in large concentrations, and it diffuses very slowly. So a plant in water can easily use up the carbon dioxide in the water immediately surrounding it, and then it has no resources with which to continue photosynthesis. However, carbonate and bicarbonate can be used to substitute for CO2 gas. So carbon dioxide that dissolves in water can dissociate into particularly bicarbonate, and plants can take in bicarbonate, convert it to CO2, and then feed it into their photosystems. Since carbonate and bicarbonate occur in much higher concentrations in water than carbon dioxide, this is one solution to the lack of carbon dioxide for photosynthesis. There's another issue, though. Even if you do use bicarbonate, you can easily exhaust the amount of bicarbonate in waters adjacent to your tissues, and then you're once again in the situation of not having any substrate for photosynthesis. Now, when you have an area of still fluids next to the surface of a living organism, we call that a boundary layer. Both CO2 and carbonate diffuse slowly, so if the water around a plant is still, if it's not moving, it can easily be exhausted in the boundary layer. So one adaptation that plants in aquatic environments have is to minimize that boundary layer. Try to minimize the extent to which water stays unmixed around their surfaces. The way you do that is by having extremely smooth surfaces that don't impede the flow of water to the maximum extent possible. So typically, aquatic plants will have very smooth leaf surfaces, which will be very unlike terrestrial plants. That encourages the free flow of water over the surface of the leaf and the replenishment of CO2 and bicarbonate. Now, oxygen is also in limited supply in water. It's 21% of oxygen. Oxygen is 21% of the atmosphere, but only about 1% in water. So if you're a mammal and you rise to the surface to breathe, or if you're a fish that gulps air, that's not a problem. You get plenty of oxygen. However, if you're relying exclusively on gills to filter oxygen from water, you need a very efficient system to extract oxygen from water. The other issue is that we know that some bodies of water can become oxygen depleted and become anoxic. And that is typical of deep waters where you don't have plants producing photosynthesis, which has oxygen as a byproduct. So deep waters can often be very challenging for animals to live in, and they may have to have very low metabolic rates in order to function in the low oxygen environments. Now, how do fish maximize the extraction of oxygen? Well, they use a method called countercurrent circulation. And countercurrent circulation in the gill means that blood flows in the opposite direction from the flow of water. That maximizes the extraction of oxygen, and it also, as a byproduct, maximizes the diffusion of CO2 out of the blood. So let's look at what the two alternatives would be: concurrent flow versus countercurrent flow. At the top here we have a diagram illustrating concurrent flow. So if the blood in the gill flows through the little gill in the same direction as water, and over here on the right we have a diagram showing water that's carrying lots of oxygen flowing from left to right. Okay. If the blood in the gill were also to flow from left to right, what you'd find is that the blood coming into the gill, first encountering the water, would be highly depleted in oxygen. And there'd be a really high diffusion gradient of oxygen out of the saturated water and into the depleted blood. But at some point, that diffusion gradient is going to decrease as oxygen travels into the blood and leaves the water. Eventually, they'll equilibrate, and you won't get any further diffusion. So when the water in the gill and the blood in the gill have equal oxygen concentrations, you won't get any further diffusion of oxygen into the blood. Okay, well, that's a system that can work, but it's not the system that aquatic organisms actually use, because countercurrent flow is much more efficient. In countercurrent flow, the blood entering the gill enters from the opposite direction of the flow of water. So the water flows from well, let's see. Which way have they got it going? Oh yeah, it's the water is flowing in from the right here on the bottom, and the blood is flowing in from the left. So here, what you've got is water that is flowing in, it's saturated with oxygen, and it's meeting blood that has been absorbing oxygen from the water all along and has increased in oxygen saturation almost to the point where it's equilibrated with the surrounding water, but not quite. Water still has a higher oxygen concentration than the blood that it's passing over. So there's a diffusion gradient, even though this blood has absorbed a whole bunch of oxygen already. As some of the oxygen is pulled out of the water as it passes through the gill, it's still meeting blood that has less oxygen in it because it hasn't been absorbing oxygen as much. So what you get is a continuous diffusion gradient. Highly oxygenated water is still giving up oxygen to nearly oxygenated blood. Partially deoxygenated water still has more oxygen than the partially oxygenated blood, so there's still diffusion of oxygen from the water into the blood. And finally, when much of the water has had its oxygen depleted by flowing into the blood, it's now meeting the most depleted blood that has the lowest oxygen concentrations. And even though the water oxygen concentration is as low as it can get, it's still got more oxygen than the blood it's encountering. So there's still an oxygen gradient from water to blood. So throughout the gill then, there's a concentration gradient that's maintained, and it's far more efficient than concurrent flow because the entire length of the gill, instead of only half of the gill, is available for oxygen diffusion. Now, organisms can survive in anaerobic or anoxic conditions, but their metabolism may have to drop. On the other hand, they may use alternative means of getting oxygen if the water is too depleted of oxygen, and that includes things like rising to the surface to gulp air if you're a fish, or if you're a plant, produce roots that extend above the soil surface to directly absorb oxygen from the air. And then finally, you can switch from oxygen-based respiration to anaerobic respiration, which typically involves something like either fermentation or perhaps sulfur metabolism. And we think that sulfur-based metabolism preceded oxygen-based metabolism in the history of life on Earth because free oxygen was not available on the early Earth. There are still some organisms in deep ocean sediments that use sulfur as the basis for their metabolism instead of oxygen. Okay, let's finish this up by talking about heat and thermal regulation in water. So proteins and other things inside living organisms need to have a stable three-dimensional structure in order to function. If they get too hot, they will lose their hydrogen bonding that keeps them in their functional conformations, and they will denature. If they get too cold, then the efficiency with which they perform their chemical reactions will drop considerably, and metabolism will be greatly impaired. So one of the big challenges for aquatic organisms is ensuring that their protein structures, specifically their enzymes, are able to function at the prevailing water temperature that they encounter. Now remember, water temperature is very stable, so this is something that aquatic organisms can actually fine-tune pretty effectively. Now you have all kinds of organisms that live in water and all kinds of protein conformations that can function in very different temperatures of water. You have some thermophiles which live in extremely hot water. Some are capable of living in thermal geysers that are at 100 degrees centigrade and periodically erupt, or even hotter if they're in the deep ocean thermal vents. The pressure of the deep ocean can result in water that's heated to above 100 degrees centigrade. So we're exploiting some of these extremophiles for their enzymes, which we use in industry and medicine. Now, more typically, we have issues of heat loss in organisms because, of course, water has a high heat capacity. It absorbs lots of heat very effectively. And so aquatic organisms that try to produce heat in their tissues will promptly lose that heat to the surrounding water unless they have special adaptations. The vast majority of aquatic organisms are what we call poikilothermic. They simply assume the temperature of the surrounding water, and their enzymes are built to function at those temperatures. However, there are a few types of aquatic organisms that attempt to maintain a higher body temperature than their surrounding water. This is very beneficial for those organisms because their metabolism is faster, they can move faster, and they can be more effective hunters, especially. So you get large predatory fish that adopt the strategy of raising their body temperature and maintaining a high body temperature compared to the surrounding water. Now, how on earth can they do it? It's ruinously expensive metabolically. So what are the adaptations that they use to reduce heat loss to the surrounding water? In most fish, what happens is the heart, which is a two-chambered heart, pumps blood out to the gills, and then from the gills it circulates not back through the heart, but directly to the tissues. So in most fish, what happens is the heart is right under the vertebra. It pumps blood into the gills, where it picks up oxygen but loses all its heat, and then it penetrates the muscles, giving up its oxygen and cooling the muscles that are generating metabolic heat. And then the veins that pick up this deoxygenated blood circulate back to the heart in the center of the fish. So it goes from the center of the fish to the periphery where the gills are, and then back into the muscles, and then back to the center of the fish. So it's a one-way circuit. In fish that have heat exchange, that maintain a high body temperature, they use countercurrent flow. They don't have a one-way circulation system. Instead, the heart in the center of the body pumps blood to the gills, where it loses all its heat but picks up oxygen. And then on the outside of the fish, as diagrammed here in the red, that oxygenated blood penetrates the muscles, gives up its oxygen, and then the blood that's lost all its oxygen is transferred to veins, collected in veins, which then don't go to the interior of the fish, they go back to the exterior of the fish, right alongside the incoming arteries. So that deoxygenated but warm blood, because the muscles have been generating heat, the blood has lost its oxygen but it's gained heat. That blood is circulating in a countercurrent direction next to oxygenated but cold blood. So that oxygenated but cold blood is picking up heat from the warm but deoxygenated blood in the veins as it goes into the muscles. That prevents the cold oxygenated blood from cooling off the muscles, and it allows the muscles to accumulate heat and maintain a body temperature higher than the water around it. Now, water is so effective at stealing heat from organisms that aquatic organisms that do this can't maintain really, really high temperatures compared to the surrounding water, but they can do pretty well. So here's the temperature trace of a bluefin tuna in water, and here it's active over a period of time, and notice that it's maintaining a body temperature of roughly 22 degrees, 23 degrees, and that's pretty constant. And look, it's diving in water that's as low as 14 degrees here in the first hour, so it's maintaining a gradient, a temperature that's up to 10 degrees higher than the surrounding water. When it dives into colder, deeper water, that gradient can be even stronger. But countercurrent flow is effective enough that big fish that have lots of insulation, like tuna, can maintain a higher body temperature than the surrounding water. It's only really large organisms that can do that, though. Okay, a couple of notes to finish up this discussion. Cold temperatures impose their own set of problems, and you don't want to be in water that's so cold it freezes you. And we know that Arctic and Antarctic fish can swim in water that is actually below the freezing point because of the pressure and the solutes that are in it. So how do you swim in water that's colder than freezing without freezing yourself? Well, once again, you can lower the freezing point of water by increasing the osmotic concentration. If you pump lots of solutes into your blood will not freeze at 0 degrees centigrade. It may freeze at -4, -6, and then you can function in these extremely cold waters. Okay? Frequently, these compounds are things like glycerol and glycoproteins, and they have the benefit of not being involved in other kinds of metabolism to any great extent, and being non-toxic. They can raise the osmolarity of blood to the point where blood will not freeze at 0 degrees centigrade. They will endure supercooling. Okay. Okay, finally, let's finish this up by talking about the idea of thermal optima. We know that water is very thermally stable, and you want to tune your enzymes to the prevailing environmental conditions. So species that live in cold water have enzyme structures that are optimized to function efficiently in cold temperatures. If they encounter warmer waters, they can actually go into heat shock and die. If you look at tropical species, they have isozymes of the very same enzymes that have a slightly different structure that works optimally in much warmer temperatures. If they encounter cold conditions, their enzymes work much less efficiently. They go into shock, or they just stop functioning and can die. But what about animals that encounter fluctuating conditions? If you have a set of enzymes that are optimized for cold conditions or a set of enzymes that are optimized for warm conditions, you can swim just as efficiently, you can hunt, you can reproduce just as efficiently in either warm or cold temperatures. But what if the temperatures do fluctuate? That would seem to be a really serious problem for aquatic organisms. Well, fortunately, enzymes are regulated by genetic loci that have 2 alleles. And a common strategy in aquatic organisms that encounter seasonal change in water temperature is to be heterozygous for different isozymes. One of the isozymes might be a form that's more effective in cold water conditions, and the other might be a form that is more effective in warm water conditions. So here's an example of rainbow trout, and rainbow trout live in freshwater rivers and streams in the northern hemisphere. that encounter winter cold and summer warmth. And sure enough, they have two isozymes. They have a winter isozyme, and here's its activity level depending on the temperature of the surrounding water. And so one isozyme is extremely efficient from 0 to 10 degrees centigrade. It drops off in efficiency above 10 degrees centigrade, but the other isozyme picks up and becomes more efficient at 10 degrees centigrade and is optimized for temperatures between 10 and 20 degrees centigrade, which is typical of the water temperatures that this particular fish encounters. So, we've covered 4 general types of general adaptive problems here. Density and viscosity, gas exchange, osmoregulation, and thermal optima. The challenges on land are quite different, and you'll see that in the next presentation.